So go ahead and put your names in. We're going to go ahead and walk through apoptosis real quick, because apoptosis is a fantastic example of a highly regulated cellular messaging process. Okay? So, when cells are infected with a virus or a bacteria, when they are damaged because you sat out on the beach too long on Cancun during spring break, right? Don't you peel? Yeah? That's because your cells were so damaged, your body said it sent the signal to commit suicide. Okay? It's to protect you. Because you guys all know that UV radiation leads to cancer, right? So if you were to keep all of those cells alive, you would be at risk for cancer. So your cells receive signals from your immune system that says, you need to die. You need to commit suicide for the betterment of the whole society. Okay? We call this process apoptosis. It's one of my favorite words in science, apoptosis, right? It's a very fun one, okay? Now, this works using mechanisms you guys already know. Which vesicle do we know that's really good at chewing things up? What is it? Lysosomes, right? We learned about lysosomes. They have enzymes in them, right? Hydrolytic enzymes. Hydrolytic enzymes catalyze what kind of reactions? Think about the name, hydrolytic enzymes. What kind of reactions do you think they catalyze? Hydrolysis? Do you remember that from like lecture 3? Yeah. Dehydration synthesis puts polymers together. Hydrolysis breaks them apart. What happens in apoptosis is that these lysosomes fuse with whole cells, whole components of the cells. The lysosomes from inside of the cell do the damage, okay? They release these hydrolytic enzymes and they hydrolyze all the cellular components. The genome gets broken down. The lipid membranes get broken down. All those sugars that were stored, they get broken down. Everything gets broken down. Now, what could have been a cell that turned into cancer, or what could have been the cell that started the outbreak of gonorrhea in your body, has been killed and destroyed. Okay? All right. So apoptosis is a very carefully regulated process, because you might imagine you don't want your cells going around committing suicide whenever they want, right? That just doesn't sound like a very good thing for your body. What might interest you, though, is to realize that this process is actually regulated here. What is that organelle? Yeah. The mitochondria has these special signal proteins. They're actually enzymes. They participate in that signaling cascade called caspases. Okay? Caspases encourage apoptosis. So we call them pro-apoptotic, right? Okay. Now, there are a variety of different signals that can regulate apoptosis. Sometimes they're internal. If something happens to your DNA, right? Let's say you sat outside in the UV too long and your DNA started to get mutated, started to change. That will actually cause a stop in cell control, and it will start to build up these pro-apoptotic signals, and once you have enough of them, they win, and the cell will die. Sometimes you want to commit suicide, right? Because the cell, the body tells you to do so. Let's say you're infected with a virus, or you're no longer needed. The body's immune system will come along and say to you, you need to commit suicide. Okay? It's a tightly regulated cascade of events. If it's a cascade of events, that provides lots of opportunities for you to amplify the signal or reverse it. Lots of opportunities to regulate it so that your cell doesn't commit suicide if you don't want it to. But it isn't always bad. I bet a lot of you appreciate the fact you have 5 distinct fingers, right? No webbing. They're not connected. They're whole. But they didn't start out that way. You started out with little nubbies. There's your little nubbies. Okay? We all started out with little nubbies. And then apoptosis received signals to start killing those cells in between what would become your 5 digits. Okay? Apoptosis played a role in how you developed by choosing very carefully, tightly regulated, which cells committed suicide and which did not. So, take a look. This is one of my favorite videos. You're going to watch this guy down here, right here. There's its chromosomes condensing down to chromatin. That is a microtubule spindle attaching to the proteins at the centromeres of those chromosomes. The microtubules are wiggling back and forth to line up, and watch, that final chromosome gets in line and boom, they divide. That's cytokinesis, the breakdown and the separation of the cytoplasm. So you can actually watch as those centrosomes form the cores of the two sides of that spindle. The chromosomes wiggling back and forth attached to the microtubules as they arrange at the metaphase plate and then are pulled apart, okay, in anaphase. And then microfilaments pinch off and separate the cytoskeleton, while those intermediate filaments reform the nuclear lamina and the nuclear membrane. All cells do this. Okay? All living things have to have this ability to do mitosis. It maintains genomes from one generation to the next. Okay? Now, most cell division on the planet is mitosis. Sometimes we get caught up in this idea of meiosis, right? And how special meiosis is for sexually replicating organisms. And it's true. Meiosis is an incredible tool for diversity and evolution. But the reality is, it's a very, very minuscule part of the entirety of cell division. The vast majority of your cells only ever see mitotic division. You only have 2 little cell types that do meiosis. Everything else in your body undergoes mitosis. All single-celled organisms, which make up the vast majority of mass, right? Your body, in terms of mass, is more single-celled bacteria and other organisms than it is you. They only do mitosis. They don't replicate sexually, okay? In order for the egg, the fertilized egg, which we call a zygote, to grow, it uses mitosis. In order for your body to repair damage, when you're an idiot and fall off your skateboard, you use mitosis. In order to grow a new bone, because you broke your bone, okay, that's mitosis. Meiosis is the exception to the rule. So you need to kind of work to think about mitosis as being your standard thought when you think about cell division. We're going to talk about meiosis. It is important. It does have its role. But mitosis, you could argue, is the more important one. Vocabulary is a big part of understanding and describing what goes on in cell division, because we have to be able to talk about what's happening to the DNA. And this is a challenging one to keep straight. So, I don't usually recommend vocab cards, right? Vocabulary cards can be a little tough, especially given the amount of vocab we have in this class. But this is one area in which I do recommend it, because you need to be able to use the right words, okay? Now, all of the DNA in the nucleus is what we call the cell's genome. Why do I specify all the DNA in the nucleus? Isn't that where all the DNA is? What else is there? Yeah, remember the mitochondria and chloroplasts? They have their own little genomes, okay? But when I'm talking about eukaryotic cells and I say genome, I'm really talking about what's in the nucleus, okay? Mitochondria replicate themselves. Chloroplasts replicate themselves the way bacteria would. Some eukaryotes organize their genome into multiple pieces of DNA. Each one of those pieces is called a chromosome. It has a set number of genes in it, okay? Humans are what we call diploid. We're going to talk a lot more about this when we come back from spring break. But what does diploid mean? Yeah. It means you have two versions of each, right? Diploid means you have two versions, one from mom and one from dad. Your mom can have blue eyes and your dad can have brown eyes, which means that your chromosomes will be a little bit different because you get a different one from mom than from dad, okay? It means you have two of each. These are called homologous chromosomes. We're going to talk more again about them in the future. So each one of those chromosomes shares a structure, right? Different chromosomes have different nucleotide sequences, but their structures are the same. All eukaryotic chromosomes, and even some prokaryotic, if you're talking about archaea, are organized around proteins. In eukaryotes, we call these proteins histones. They're cool. Histones are really cool. You guys are going to like these when you get into genetics, okay? You actually wind your genome around. Like this. See here? You actually wind your genome around these proteins. And it condenses it. It organizes it. Ever hear that each person's genome could stretch like to the moon, if you were to stretch it all the way out? And yet it fits in your body. And it stays organized. It doesn't tangle up into a knot because it's organized around these histones. In preparation for DNA or for cellular division, your DNA condenses. These histones further condense around each other, okay? They actually twist into these structures that we call chromatin. This only happens during division, okay? This only happens during division, that we condense down into this very dense chromatin. And you saw that in that video that I showed you, right? We went from this big kind of diffuse red nucleus with all the DNA to all of a sudden having these very distinct structures. Before we go through this process though, right? Before we go through this process, we have to replicate the genome. When we replicate chromosomes, we call that pair of exactly identical chromosomes sister chromatids. They are exactly identical, okay? Is the chromosome from your mom and the chromosome from your dad, are they sister chromatids? No. Those are what we call homologous chromosomes, okay? It's another one of those. Make sure you keep those two straight. They're a tough one. Once these replicated chromosomes, called sister chromatids, prepare for DNA division by condensing into chromatin, they show up as these very iconic X structures. We call this waste right here, okay? We call this waste the centromere. Not the centrosome, the centromere, okay? The arms are held together by proteins called cohesins, right? They're cohesive. They hold sister chromatids together, so we call them cohesins, okay? Another group of proteins is found at these centromeres, and they're called kinetochores. When you see that kinet, K-I-N-E-T, what word does that make you think of? Like kinetic energy, right? Which is like active, moving energy. Kinetochores are going to be where these spindle fibers actually attach to move the chromosomes. Okay? Hence their name kind of revolves around this idea of movement. All right? Just like how cohesins hold the two sister chromatids together cohesively, kinetochores will be part and parcel to the moving of those chromatids. Here's what it actually looks like. Here's the chromosomes in blue. They have condensed into chromatin. You can kind of start to see some of those arms here, those organized structures. The kinetochores are in green. Those are found at the centromeres. Okay, the kinetochores are at the centromeres. The microtubule spindles are shown in red. They attach the kinetochores at the centromeres to the centrosomes. It's a lot of C and K words, right? A lot of very similar sounds. But you've got to be able to work on keeping these vocabulary words straight. All right, so the human genome, right? And we are talking about somatic cells here, not gametes, not egg and sperm, everything else. Literally everything else in your body is a somatic cell. Mitosis is the rule, not meiosis. Somatic cells are the rule, not gametes. You have 23 pairs of chromosomes. One from mom, one from dad. Those are your homologous chromosomes. They're the same chromosome, just two versions of it, right? Homologous means same, okay? Two of the same, all right? But it's one from mom and one from dad, so they can vary a little bit. They might not be 100% identical. Sister chromatids, which are when you replicate mom's chromosome and you replicate dad's chromosome, they're identical. Okay? So here is one chromosome, say from mom. Here's a second chromosome from dad. These are homologous chromosomes. When they duplicate, each one forms a pair of sister chromatids that is identical. All right? Now we're going to talk about how these divide, okay? Because mitotic division is essential for maintaining the amount of genetic information in each cell. Too much or too little is a very bad thing, okay? The cell cycle is a very closely regulated series of events. And you need to know what happens in each step and how we regulate them. Interphase can be either resting or growth. It's what happens between mitotic divisions. Okay? Most cells in your body are actually at some point in interphase for the vast majority of time. Okay? Mitotic phases are going to include the traditional stages of mitosis as well as cytokinesis. How many of you guys have heard of cytokinesis? That normally taught with mitosis? Awesome. Cool. We're going to go through it again though. So interphase, which is about 90% of the cell cycle, it can be divided into three major phases. The first is G1. G stands for gap, by the way. But it also is a phase in which the cell grows. The vast majority of cell growth actually occurs in G1. Okay? As soon as the cell has divided, because remember, when we are in G1, we are just getting out of our prior cell division, entering into a new G1 phase. So when we're in G1, we just divided, and the cells are going to about double in size. Now the cell has to make a decision. It has to decide if it's going to divide again or not. If it's a non-dividing cell, it will go into this long-term resting phase that we call G0. Cells that are in G0 just stop. They don't go any further. After they've grown from their division, they just go into doing what they normally would do. Okay, most of your cells do this. They just go to G0. But the cells that are going to divide again need to prepare for that division. So they will move from G1 into S phase. S stands for synthesis. This is where your chromosomes are duplicated. Chromosomes are duplicated in interphase, specifically S phase of interphase. Mitosis has nothing to do with DNA replication. Once you synthesize new DNA, once you now have sister chromatids, you can go on to the second gap phase. This is when, like, mitochondria divide, chloroplasts. You can synthesize necessary energy that's going to be needed to undergo this highly energetic process of cellular division, all of those kinds of things. Okay? It's like a prep phase. So mitosis is conventionally broken down into 5 stages. Prophase, prometaphase, metaphase, anaphase, and telophase. Okay? Did you guys do prometaphase in school? Some yes, some no. It falls right between prophase and metaphase. Okay? Now, I know that many of you guys like the ability to be able to kind of remember some of these things with acronyms. So here's how I remember it. PPMAT. PPMAT. Okay? This is PPMAT. And it is not a PPMAT unless you have your second P, so don't forget prometaphase. Okay? This is how I remember it. PPMAT. There is a full spread, like 2-page spread that walks you through all of these steps in your textbook. Okay? So we're going to walk through them. Okay? We're going to talk about what happens at each step. But if you need to go back and look through them, that's all there. G2 is the last phase of interphase. This is what a cell looks like right before it goes into mitosis. The cell has already doubled in size. What phase was that? The cell has duplicated its chromosomes. What phase was that? The cell has duplicated its centrosomes. What phase was that? S phase. And finally, the cell has prepared to divide. It's duplicated its chloroplast, if it has it. It's duplicated its mitochondria. It's made lots of ATP. What phase was that? G2. So that's what's happening right now. Note that those chromosomes are duplicated, but they haven't condensed yet. They're not chromatin yet, okay? It's only in prophase, the very first mitotic phase, that we see them condense down, okay? Now, those centrosomes are going to start to spread out. They spread because their microtubules come together and actually push them apart, okay? They're going to spread out, but the nuclear envelope is still intact, so we don't have any attachment of the microtubules to the kinetochores. That happens when we get into prometaphase. Prometaphase is when these microtubules have attached to the kinetochores because the nuclear envelope has started to fragment. It's started to break up. Okay? One of those chromosomes is from mom and one is from dad. They're homologous chromosomes. Each one has its own pair of sister chromatids. And watch for them, because the sister chromatids are going to be the ones that split. In metaphase, this almost instantaneous phase, it's very, very, very short, but it's very important. Each one of those chromosomes has a microtubule on both sides. attached to the two kinetochores on either side of the waist. And they're pulling like this, back and forth, back and forth, back and forth. You might remember in that video, it wasn't until every chromosome was lined right up down the middle that the cell actually split. Metaphase is when we form that metaphase plate and the chromosomes align because of that tugging. In anaphase, what happens is that, again, microtubules push the centrosomes apart. And at the same time, those chromosomes walk along the microtubules. The cohesins, the proteins that held the sister chromatids together, dissolve, allowing them to walk backwards. Okay? We now call them daughter chromosomes. So you may see that terminology. They're not chromatids anymore because they're not attached to one another. So they kind of go back to being like a whole chromosome in their own right. So sometimes we call them daughter chromosomes. All right? What happens is, as that cell gets pushed apart and the cell elongates, it provides the space needed for the new nuclear envelope to form and the cytoplasm to be pinched in half. That's called cytokinesis, okay? And cytokinesis occurs when these actin filaments form like a string that gets pulled taut, pinching between them. And I'll show you guys a picture of that in a minute. At the end, here is mom's chromosome and dad's chromosome. Note that each daughter cell has one of mom's and one of dad's. We've created two genetically identical daughter cells. Okay? They have the same DNA as the parent cell, and they themselves are identical to each other. All right. So this is what it looks like when you put it all together. And I'll post a video for you guys to watch, okay, so that you can watch through this whole process. All right? But when you have prophase here, this is when those chromosomes begin to condense into chromatin, and our centromeres begin to push out, forming a mitotic spindle. In prometaphase, the nuclear envelope dissolves, allowing those microtubules to attach to the centromeres via kinetochores, proteins. In metaphase, we line all of our chromosomes up along the metaphase plate because our spindle fibers, the microtubules, pull and push until they line up perfectly. Then the cohesins that hold the sister chromatids apart, they dissolve and allow the sister chromatids to separate, forming new daughter chromosomes. Each one is identical to the one before it. And they're going to be pulled back to the poles of these spindle fibers. Okay? And then once these have pulled apart, the cell can divide, forming two new nuclei. Each one has its own centrosome, its own network, and it'll be pinched in half. So here's one mom's chromosome, one dad's chromosome. Here they split, and you're left with one in each. All right, so we're going to take a closer look at some of these formations. The mitotic spindle, for example, right? This is that network of microtubules. They originate at centrosomes. Those centrosomes got duplicated during S phase at the same time that we duplicated our DNA. In prometaphase, those spindles actually attach to the kinetochores of centrosomes. Sometimes they'll also touch others, right? Other spindle fibers, okay? In metaphase, that push and pull of the kinetochore-bound microtubules will line up chromosomes on the metaphase plate. And then in anaphase, we're going to split sister chromatids using the kinetochore-bound microtubules, but we're also going to push apart using those non-kinetochore-bound microtubules. Okay? No, it doesn't like me today. I'll post it for you guys. But it looks like this. Here's your centrosome here. And you can see the vast network of microtubules that are extending outwards, right? Some of those microtubules will actually connect to these kinetochores that are bound, that are these centers. You can see here's a way zoomed in scanning transmission electron of these kinetochores, these proteins on the centromeres, and the microtubules that extend from them. Okay? There are motor proteins on those kinetochores. Okay? That's how these chromosomes, these sister chromatids, separate and walk in opposite directions. So if you have, here's an example of a chromosome attached here on the end, right? There are, in that kinetochore, motor proteins. Those motor proteins are going to walk backwards, like this. It's not microtubules pulling. It's actually chromosomes and the motor proteins in their kinetochores that walk themselves backwards. And then from the other end, those microtubules depolymerize. Okay? So what I want you guys to take away from this is the importance that these kinetochores have motor proteins. These are active. They're actually walking themselves backwards along these microtubules. Okay? If you go in your textbook, you can read about this experiment. It's a very cool one. They actually mark the area, okay? They actually mark an area of this spindle to show that the spindle doesn't pull backwards. The microtubules walk backwards, and then the spindle depolymerizes from this end, okay? Cytokinesis is fun because it makes cells look like butts. There you go. You'll always remember it now, though, right? Cytokinesis makes them look like butts. And it's got a great name. It's called a cleavage furrow. Okay? Yeah, it's called a cleavage furrow. So, this is formed by microfilaments. Microfilaments tied like a string around the cytoplasm. What do we call the microfilaments that form around the outside of the cell? Starts with a C. The microfilaments around the outside of the cell. That's the cortex, yeah. That cortex is actually going to condense downwards. And when it condenses downwards, it's going to act like a string, pinching the cytoplasm in half. So you can see here how you actually get this condensing, right? This condensing area. And it's going to pull these until they become two separate daughter cells, okay? This is how microfilaments participate in mitotic division. The sequential events of the cell cycle are directed by a series of control steps. So these control steps are going to be times in which during the cell cycle, the cell is going to check itself. It's going to check the status of its DNA, its location, and its quality. It's going to check its external environment and its internal resources to decide whether or not it should be proceeding to the next step in this cell division cycle. There are multiple different checkpoints, but we're only going to talk about a couple of those checkpoints. The mitotic checkpoint decides whether or not the cell can actually complete mitosis and divide into two independent daughter cells. The G1 checkpoint is an opportunity for a cell that's not going to be dividing again to actually exit this cycle into that G0 phase that we talked about, the non-replicating phase. And this G2 checkpoint here makes sure that before a cell actually goes into mitosis, that it's met all of the necessary requirements and that it's prepped and ready to go. So let's take a look at an example of one of these checkpoints. When a cell is in mitosis, okay, it's going to need to make sure that it's past the G2 checkpoint first. And it begins the process of attaching those microtubule spindles to the kinetochores, the proteins that are at the center, those centromeres of the chromosomes. until every one of those kinetochores has a microtubule spindle attached. They're going to be sending out stop signals to the cell. Only when every one of them has attached and they line up on a metaphase plate do they actually send the go-ahead signal for there to be division of those sister chromatids, the cohesins dissolving away, and the separation of the chromatids that we call anaphase. So a cell controls whether or not it proceeds to the next step by ensuring that it's met all of the necessary checkpoints. Another example is this G1 checkpoint. When the G1 checkpoint happens, there has to be a signal, and it's typically an external signal that tells the cell, yes, I want you to divide. It's a way of protecting the cell against things like cancer. So if you don't have this signal to divide, you don't ever proceed past this G1 checkpoint, and you go into that phase that we call G0. It's a persistent, non-dividing state. It's actually what most of your cells are sitting in all their lives. Now, there are these signals that are dependent specifically on what the internal environment of the cell looks like, such as whether or not all spindles have attached to the kinetochores. But there are others that are dependent on concentration. Meet cyclins. These are proteins whose expression is dependent on the phases of cell cycle. You can see here that cyclins increase in concentration, peaking during mitosis, and then are dissolved away. They actually get broken apart as the cell leaves mitosis. As you enter that G2 phase again, the cell will increase the concentration of cyclin protein until eventually it collapses and it leaves mitosis. These cyclins act in concert with kinases, and we call these cyclin-dependent kinases because the activity, the enzymatic activity of those kinases is directly dependent on the cyclin concentration of their partnered cyclin. You can see here in orange that even when the cyclin concentrations are at its highest, that's the only time that you see any kinase activity of those cyclins. And that's essential for moving through that mitotic checkpoint and into the next phases of interphase. And as that cyclin is degraded, you also see the degradation of the kinase activity. So it looks something like this, where you have synthesis of these cyclins and accumulation that's especially high during G2, and this actually allows the cell to pass its G2 checkpoint and enter into mitosis. Now these cyclin and CDK complexes can actually achieve their function, and the cyclin becomes degraded in the process until we go through the process once again, and you get re-expression and reaccumulation of the cyclin. So we do have a lot of internal signals, things like DNA damage and proper spindle attachment that are really important. But I've also mentioned to you the idea that especially for that G1 checkpoint, but for others as well, there are external factors. An example of one of those external factors are growth factors, and they do just what you think. These growth factors stimulate a cell to grow and divide. These are going to be examples of endocrine signaling factors, things that are very type specific. So for example, a bone growth factor is only going to bind to growth factor receptors on bone cells. There's also platelet-derived growth factors that will only bind to certain types of blood cells. There's a lot of different specificity here so that we can decide exactly which cells we want to grow. And that's really important because when you have cells that respond too well to these growth factors, they can grow to such levels of abundance that it's actually detrimental. This is cancer cells, cells that respond too well to growth stimuli. But there's a second component here. You also have to ignore the stop signals, right? A growth factor is a go-ahead signal, but we also have stop signals that are meant to keep these processes in check. The stop signals have to be ignored by cancer cells. And an example of one of these stop signals is density-dependent inhibition. It means that when the cells get too densely packed, they will actually inhibit themselves. Crowded cells stop dividing. Unfortunately, cancer cells ignore this, and so they keep growing and keep growing and keep growing to form what we commonly know as tumors. Now, cancer cells are your cells, right? They are your cells that just keep dividing and dividing and dividing. And the reason that they keep dividing and dividing is because they're broken. Specifically, their genome, their genetic material has been mutated. It has been changed in some way. And those changes change the way that cancer cells respond to cell signaling events. They over-respond to signals that tell them to grow, and they under-respond to signals that tell them to stop growing. And it's changes in the genetic material that specifically cause that. So carcinogens are chemical agents that mutate DNA. So you guys already know a lot of examples of carcinogens. These can be things like tobacco smoke. That's why when you smoke tobacco, it gives you cancer. Another one is UV light. Again, laying out in the sun in Cancun can give you skin cancer because you are exposed to that UV radiation. Can you think of any other ones? Now, apoptosis, that process that we talked about at the beginning of the lecture, is really critical because when apoptosis works correctly, it causes these precancerous cells to commit cell suicide, and it protects you, okay? But eventually what happens is enough of the cell's genome becomes mutated, multiple rounds of mutation. You can see here a first, a second, a third, and a fourth mutation eventually leads to cells that are simply so abnormal that they don't respond to any of our normal checkpoints. Mutations in cancer cause them to evade normal mechanisms by things like making their own growth factors. So these are going to be a type of autocrine signaling where you produce a growth factor that you respond to all by yourself. It can also cause receptors or signaling molecules to become constitutively active. That means that they're always on. You actually saw an example of this when we talked about cholera and the cholera toxin making G proteins constitutively active because that GTP stays bound. This can happen in a lot of different ways in the cell, like receptor tyrosine kinases staying phosphorylated and not responding to the dephosphorylation. Another is you can inactivate stop signals or controls. So you can actually get rid of certain kinds of caspases, or you can go through the process of mutating a variety of different proteins that serve as those stop signals. Chemotherapy drugs often act by inhibiting cell division. Since cancer cells divide very, very rapidly, chemotherapy drugs predominantly influence the division of those cancer cells. Unfortunately, a number of your cells also divide very rapidly. Not many, but some. Examples include your hair follicles, which is why your hair falls out, as well as your stomach lining, which is activated very frequently and has to replenish itself very frequently. This causes you to have a lot of intestinal and stomach distress, like vomiting, and also your nail beds, the growth of the nails on your fingers and on your toes. So this is where these effects of chemotherapy come from. It's not the cancer that makes you lose your hair. It's the drugs inhibiting cell division that cause these. Now, the real problem is that there are thousands of these different mechanisms and even more mutations that can cause each one of them. So even though we can target drugs like chemotherapy drugs to inhibit all types of cell division, this is not an ideal response. We just talked about some of those massive side effects, but they also are really damaging and they don't really cure you of cancer. So there are a lot of different mechanisms, and the field of genetics and cancer study has really been moving towards the idea that the better we understand how each of those mechanisms occurs, we can better target our medicines to fight them. So personalized or precision medicine is the idea that within a patient group, even if every single one of them had breast cancer, they cannot all be treated by the same drug. It will work for some, but others it may be toxic without even doing any benefit. Others it may benefit without toxicity, and others it may see no effect at all, because the reason why their cancers are cancerous is simply different for each. Now, you guys have heard these words before, and since we are talking about cancer, I want to make sure that you all understand exactly what they mean. When cancer cells begin to divide within a tissue, we call it a benign tumor. So here's an example of a tumor that has stayed localized within its tissue. Immune cells will attempt to eliminate them, but when they can't do so, they begin to form a true solid mass. We only call them a malignant tumor when they begin to invade the surrounding tissues, and this is a point at which it begins to cause some damage to the cell and to the body. When those cells break away and spread through either your lymphatic vessels or your blood vessels to other parts of your body, they can start to form tumors in places that they didn't even originate from. And this is what we call metastasis. Individuals whose cancers have metastasized now have tumors in more than one area of the body. If you guys are interested in learning more about cancer, these are two of the books that I highly recommend. The Emperor of All Maladies is not only a book that has a fantastic review of cancer, and it writes in very plain English, but it also talks about the latest medicines. It's also a PBS documentary, which you can watch for free using your local TV subscription online. It's a 3-part documentary and has lots of excellent cancer stories, survivor stories, clinical trials, and again, it's all in a very basic science, right? It's from PBS. The other is The Immortal Life of Henrietta Lacks. This tackles the morality of cancer rights and research. It's a relatively new book, and it was turned into an HBO movie, although I admit that I haven't seen it. But the book is a very, very easy read, and it was actually written so that high schoolers could kind of understand what happened. It's the story of a woman named Henrietta Lacks, whose cancer cells were harvested, and they became the first human cells ever to grow outside of the body. They still grow today, and I've even used them in my own research. But it was her story alone that led to the idea of patients' rights. And for anyone who's interested in going into fields of medicine, I highly encourage that you check out this excellent book. Here is a review of today's topics and some of these concepts that we've worked through. All of these slides are available to you in the same place that I post all of them. If you guys have any questions, please do not hesitate to reach out to me by email, and I will be available over spring break to help you work through some of these if it's needed. All right, guys, enjoy your spring break, and thanks for listening.